Welcome everyone to today's Exploring Blackleg Management Tools and Quantitative Resistance workshop, brought to you by the Canadian Phytopathological Society. This workshop is the third out of a series of 4 post-Tri-Society virtual workshops this year, which you can find on this website. The CPS invited Gary Peng and Delantha Fernando to organize this event, along with Justine Cornelissen, who helped in the background from the Canola Council of Canada. Today, I will give a brief overview of the Blackleg research topics in the Canola Agri-Science Cluster that is part of the Canadian Agricultural Partnership Program. The Canola Council of Canada and SAS Canola are administering Theme 7 of this cluster, which has several Blackleg and verticillium projects. There are 2 projects in the canola cluster that are working on developing protocols to measure quantitative resistance to blackleg in canola. And being able to put a number on the level of QR will enable breeders to more effectively screen for QR in the breeding programs. In the future, it'll also help benefit the canola industry by allowing seed companies to develop and include a QR level on their variety labels to complement basic blackleg resistance labeling, as well as the more comprehensive major R gene labeling system in Canada. Research in this cluster is looking at the critical infection window of blackleg in Western Canada. And due to the short growing season, significant yield loss from blackleg usually doesn't occur unless there is early infection of the cotyledons or the first few true leaves. A separate activity is looking at the possibility of early infection via flea beetle wounding and whether seed treatments or other flea beetle control has an effect on blackleg incidence and severity. And another project is evaluating the stewardship of R genes in canola to determine what is the best order to deploy different R genes in varieties to maintain the durability of major gene resistance. And finally, genes within the RLM 3479 R gene cluster are being cloned to study the genetics of the interaction with avirulence genes. And this may lead to better R genes being developed in the future and possibly creation of new R genes that can detect new blackleg races that evolve. And just to give you an idea of some of the knowledge transfer products that have been developed from this work so far are this new blackleg disease life cycle infographic, as well as this new blackleg yield loss calculator. And both of these products can be found on the Canola Council of Canada's website. And Justine will be discussing them in more detail later today. So with that, I thank you for your attention and enjoy the talks today. Thanks.